certain so-called mitochondropathies, diseases like Kearns-Sayre syndrome and chronic progressive external ophthalmoplegia, can be effectively treated with coenzyme Q10, rendering these individuals with less lactate and pyruvate levels following exercise, and that's a clear indication of mitochondrial upregulation. Well, Dr. Lest, writing in Free Radical Biology and Medicine in 1999, found that there is an inverse relationship between coenzyme Q10 levels in the mitochondria and superoxide or free radical generation. An interesting curve here that looks sort of reminiscent to one we've seen earlier. And here we find that higher levels of CoQ10, as we would see in pigs and cows and rabbits, are associated with less free radical generation of the superoxide type. That animals having less coenzyme Q10, rats and mice, for example, have shorter lifespans and, in fact, have higher generation of superoxide. And here is the previous curve. Again, it makes one wonder if, indeed, lifespan may be directly related to availability of coenzyme Q10. Well, Dr. Schultz, working in Dr. Flint Beal's lab at Massachusetts General Hospital, publishing in the Annals of Neurology, has demonstrated interesting deficiencies of coenzyme Q10 in patients with PD, on the left side of the curve, or Parkinson's disease. There's a profound reduction of available coenzyme Q10 not only in the Parkinson's patient, but interestingly in the spouse as well, and this certainly makes one consider that perhaps there is some environmental influence lowering available coenzyme Q10. Well, the question then is, does repletion of coenzyme Q10, or does the deficiency, rather, of the coenzyme Q10 relate to the deficiencies of mitochondrial function, specifically of complexes 1 and 2,3 that these individuals manifest? And in fact, there was a very good correlation. As they stated, lower levels of coenzyme Q10 and the activities of complex 1 and complex 2,3 were significantly correlated. So the question then becomes, what is the effect of the low coenzyme Q10? And what we know is that low enzyme CoQ10 reduces complex 1 activity of mitochondrial function, which then, because mitochondrial function is deficient, enhances the production of free radicals. So we see a direct relationship between low CoQ10 and the production of free radicals. Dr. Beal's lab further has demonstrated that we can evaluate plasma coenzyme Q10 levels, and at higher levels of oral supplementation, plasma CoQ10 levels demonstrated in the yellow bars are substantially elevated. So coenzyme Q10 becomes a very important focus of our attention in terms of the activity of mitochondria. We want to identify things which can reduce coenzyme Q10, and there are certain things to be aware of. This study published in the Molecular Aspects of Medicine back in 1997, demonstrated a 19% reduction of coenzyme Q10 in individuals who are taking lovastatin, a popular cholesterol-lowering drug, and a 29% reduction of CoQ10 in individuals taking another cholesterol-lowering drug, pravastatin. So it's important to recognize that these drugs that we are using in some of our patients may have profound effects on mitochondrial function and hence increase free radical production. Further, we know that lovastatin does affect psychological well-being. In this study published in the American Journal of Medicine in 2000, it showed that placebo subjects improved in neuropsychological tests of attention with time, while the lovastatin group, or those receiving that HMG-CoA reductase inhibitor, actually declined. So again, it's very important to look at all the factors that can affect mitochondrial function, and certainly we want to take a look at some toxic events that may compromise the ability of the mitochondria to produce ATP. Now here's an interesting report coming from Neurology back in 1993 looking at the rat model, showing that levodopa actually causes a significant reduction in complex 1 activity. And as they stated, dosage comparable to that used in humans. The effect was reversible. So levodopa, really the standard drug used for Parkinson's disease, actually compromises mitochondrial function. Something very important to think about. Levodopa is, in fact, a mitochondrial toxin. Well, let's look about the effects upon mitochondrial function when they are damaged by oxidative stress. In this case, rat mitochondria were treated with ascorbate and iron to produce an oxidative stress, and we evaluate the functions complex 2, 3, and 5, and the effectiveness of various interventions. Here we see that in the control, when we introduce the ascorbate iron, shown in the red compared to the green, there's a dramatic loss of complex 2 activity. When the neurons are pre-treated with vitamin E, there is some preservation of activity with idebenone, or a brain-soluble coenzyme Q10, again a modicum of preservation of mitochondrial activity, but with glutathione, a dramatic preservation of the way mitochondrial function when challenged by this oxidative situation. Similarly, when we look at complex 3 activity, we see that there's a profound reduction when challenged by the oxidative situation. Vitamin E is very preserving in complex 3 challenge. Idebenone, or soluble coenzyme Q10, also preserves function. And once again, glutathione seems to be preserving function when these mitochondria are challenged. And finally, when we look at complex 5 activity, we see a similar display where there's a significant loss of activity in the control group when challenged by the oxidative milieu. Not much improvement with vitamin E or idebenone, which is coenzyme Q10, but again, profound salvaging of mitochondrial function by glutathione. Well, this deficiency of glutathione has not gone unnoticed. The study that is really prototypic has been the 1986 study by Dr. Perry, in which he found a profound deficiency of glutathione in the substantia nigra of patients with Parkinson's disease. And this is really an exciting article, and to the extent that he felt that was in fact the cause of Parkinson's disease. But nevertheless, despite the fact that we know it is not the end-all cause of Parkinson's, it's still a very important finding. Therefore, enhancing glutathione, schemes to enhance glutathione, are important. We know that alpha-lipoic acid is critical in maintaining glutathione levels and in fact recycling glutathione. We also know that sulfur-rich amino acids are important in regeneration of glutathione, and these include things like N-acetylcysteine, L-cysteine, methionine, glutamine, and even oral glutathione itself. Reducing xenobiotic challenges is an important factor, because we know that xenobiotic challenges which affect cytochrome P450 activity can in fact consume glutathione. And finally, we know that complementary antioxidants are critical, because all of these brain-important antioxidants recycle each other, and we certainly don't want to have any weak link in the chain. Glutathione can be retained by using milk thistle or silymarin. Let's look at alpha-lipoic acid, a very exciting antioxidant with respect to the nervous system. It is a potent antioxidant, does scavenge free radicals. It serves as a metal chelator, chelating iron, copper, and cadmium. In addition, as I indicated, it regenerates endogenous antioxidants like vitamin C, E, and also glutathione. It has profound ability to cross the blood-brain barrier, obviously proving very important for our concerns today. Now, what else activates microglia? Dietary fat, a very important consideration in our modern society. We know that dietary lipids and antioxidants, when evaluated in Parkinson's disease, it's been demonstrated that those individuals in the highest quartile of consumption of animal products have a 500% increased risk of having Parkinson's disease. Now, this is a very important consideration. Here is a dietary interaction with possibly a genetic risk factor. In this case, demonstrated to increase the risk of Parkinson's disease by some 500%. Well, why would that be? Well, aside from the fact that we know that many animal products that we consume today are laden with pesticides, on a more fundamental biochemical level, we know that diets which are rich in meat, eggs, and high in fat and alcohol do ultimately enhance the production of prostaglandin 2 series. And as we know, this is a scenario that will increase brain inflammation. And as you recall from the beginning of this videotape, we discussed how important inflammation is now being looked upon as having a causative role in these neurodegenerative conditions. Vegetarian diets, on the other hand, high in the essential fatty acids, the omega-3 and the omega-6 group, tend to produce the prostaglandins 1 and 3 series, and these tend to reduce inflammation. So these dietary choices that we make, on the left choosing a diet rich in animal products versus a vegetarian diet, do have a very important role to play in allowing genetic expression, in allowing us to either have or have not those diseases which we may be predisposed to have.